Sometimes we need data to perform our analysis, whether it's for RNA-seq studies, microbiome analysis, or other complex datasets. But what if we don't have real data readily available? Can we simulate and create data from scratch that mimics real-world scenarios? Let's see how can we generate synthetic datasets in R. Firstly, we upload our libraries. We will use PHYLOSEQ and ggplot2. So let's run our code. Then we import our data. So we write data and then we put global patterns. The global patterns dataset in R is a well-known dataset for the PHYLOSEQ package that contains microbial community data from a variety of environments. It includes samples from different sources, such as human body sites, fresh water, soil, and others. In this example, we are going to build a random OTU table as a pretend dataset, and later we can merge different data types into a single PHYLOSEQ object for analysis. So firstly, let's create a pretend OTU table. So we create a variable called OTUMat, and then we write equal to matrix sample, and then we put from 1 to 100, and then 100 replace equal true, in row equal 10, in col equal 10. Let's run our code. And here we have it. So as you can see, here in this code, we put sample 1 to 100 to generate 100 random numbers between 1 to 100, allowing duplicates. And then the matrix in row equal 10, in col equal 10, to convert these 100 numbers into a 10 by 10 matrix, 10 rows and 10 columns. So as you can see, we have here 10 rows and 10 columns. And now, so as you can see, we've created a pretend OTE table called OTUMat that mimics a table of species abundance data, like one you might read from a file in a real-world microbiome project. The table consists of 10 rows and 10 columns. Each row represents a OTU, or operational taxonomic unit, here, which is a proxy for different species in microbial community. And for the columns, each represents a sample, where the numbers in the table indicate the abundance or count of each OTU in each sample. We filled this table with random numbers, simulating what an OTU abundance table could look like. Now we need to give our matrix meaningful labels for rows and columns to represent the microbial data correctly. We will label the rows as different operational taxonomic units or OTUs, and the columns as distinct samples. So we write row names, and then we put OTUMat equal to, and then paste 0, OTU 1 to the number of rows, OTUMat. And for call names, OTUMat, we do the same, paste 0, but here's a sample from 1 to the number of columns, OTUMat. Let's run our code. And here we have it. The OTU stands for Operational Taxonomic Unit. As you can see, we have different samples. Each column represents a sample, and different rows. Each corresponds to OTU. So we have OTU 1, 2, 3 until 10. The OTU stands for Operational Taxonomic Unit. In microbiome studies, it's term used to classify groups of closely related microorganisms based on their DNA sequence similarity. Since it's difficult to define species in microbiome datasets due to genetic variations, OTUs serve as a practical way to group organisms that are similar enough. So when we refer to the OTU in the data, we are talking about clusters of microbes that are grouped together because their genetic sequences are very similar. Now we need a pretend taxonomy table. So we write taxmat equal matrix, and then we put sample letters 70, replace equal to row, in row equal in row, OTUMAT, and in column equal 7. And then row names taxmat equal to row names OTUMAT. And then call names taxmat, and we put the names. We have domain, phylum, class, order, family, genus, and species. Here we put letter 70 to generate 70 random letters with repetition. This matrix is filled with random letters and has number of rows equal to the number of OTUs and 7 columns. And regarding here, matrix in row equal row or in row OTUMAT and in col equal 7, here we create a matrix with rows equal to OTUMAT's rows or OTUs and 7 columns or taxonomy ranks. And then row names taxmat equal to row names OTUMAT to set row names to match OTUMAT, for example OTU names. And call names taxmat, here we assign a taxonomic rank names from domain to species. This step organizes the OTUs by different levels of biological classification, such as domain, phylum, class, and others. Now we have created two matrices, AutoMat and Taxomat, which represents the operational taxonomic unit or OTUs and their corresponding taxonomic classifications. This is essential because these matrices serve as the foundational components needed to build a PHYLOSEQ object. So firstly, let's check their types. So we check the type by using class. So class AutoMat, let's run our code. We got matrix array. And class taxmat, we got also matrix array. So now we are going to create the OTU and taxonomic tables for our microbial data, which are essential for further analysis in PHYLOSEQ. We do this by using the OTU table. So we put OTU table and then inside we use the OTUMAT and then taxa or rows equal true. This creates an OTU table. This table contains counts of operational taxonomic unit or OTUs, which are groups of closely related microorganisms. In this case, the rows represent different OTUs, while the columns represent different samples. And next, we make a variable tax and we put tax table taxomat. Here we create a taxonomic table. This table provides hierarchical information about the classification of each OTU, such as its domain, phylum, class, order, family, and others. The taxonomic information helps in understanding the diversity and composition of microbial communities. So let's run our code. So as you can see, we have 10 taxa and 10 samples. Here the columns are the samples and taxa are the rows. OTU1, OTU2 until 10. Now let's combine our operational taxonomic unit, OTU table and taxonomic table into a single PHYLOSEQ object, which we will call it PhySeq. So also look at taxa. So as you can see, instead of the samples, we have the domain, phylum, class, order, and here's the OTUs. And now, let's combine our operational taxonomic unit, OTU table, and taxonomic table into a single PHYLOSEQ object. So, we write PhySeq equal to, and then PHYLOSEQ, we put the OTU table and tax table. Let's run our code. So as you can see, the PhyLOSEQ class experiment level object here. This indicates that PhySeq is a specialized object designed to handle and analyze microbiome data within the PhyLOSEQ data frame. An OTU table here. This part describes the OTU table components of the PhySeq object, highlighting we have 10 taxa and 10 samples. In addition, tax table we have 10 taxa by 7 taxonomic ranks. So it refers to the taxonomy table, which categorizes 10 OTUs according to 7 taxonomic ranks, which are domain, phylum, and others. So what does PHYLOSEQ do? PHYLOSEQ combines these two tables into one object, so you can analyze them together. It's like putting how many table and the family tree into a single folder. When you look at PHYSEQ OTU table, tells you there are 10 types of bacteria across 10 samples, but for taxonomy table, shows that each of the 10 bacteria has 7 type of classification. But why it's useful? Well, it's useful because it makes it easy to study the microbes, how many are in each sample, which ones are closely related, and what trends or patterns exist. So now, let's create a stacked bar plot of the microbial composition from the PHYSEQ object. So we write plot bar and we put PHYSEQ, fill, family. Let's run our code. So as you can see, family is the letters that we put. Each letter corresponds to color. We have the x-axis, which are the samples from 1 to 10, and the y-axis is the abundance. So we have different colors assigned to various families of microbes based on the family classification from our taxonomy table. Now, let's create random sample data to add to our PHYLOSEQ dataset. This data will consist of two variables, location and depth. The location variable will contain random letters from A to D, while the depth will have random values between 50 to 1000. So let's make it. We will put sample data equal to sample data and data frame. And then we will create the location same as we said, sample and then letters from A to D, so 4, and size equal to n samples, PHYSEQ, and replace equal to, regarding the depth, samples from 50 to 1000. And then size equal n samples, PHYSEQ, replace equal true, and for the depth, we put sample 50 to 1000, size equal n samples, PHYSEQ, replace equal true, pro.names equal sample names, PHYSEQ, and strings as factors equal false. And then we write the sample data. Let's run our code. So as you can see, we have that in samples, location from A to D, A, B, C, D, and the depth from 50 to 1000. Now let's create a random phylogenetic tree with the ape package and add it to our dataset. Make sure its step labels match your OTU table. So we will write library ape and then we put random tree equal r3 in taxa PHYSEQ, rooted equal true, tip.label equal taxa names PHYSEQ, and then plot random tree. Let's run the code. And here we have it. So as you can see, we got an output of graphical representation of a random phylogenetic tree. Each branch represents a hypothetical evolutionary relationship among the taxa, with the tips labeled according to the taxa names from the PHYSEQ object. So we have OTU8, OTU5, 7, 9, and so on. Now let's combine these all together. We can do this either by adding the new data components to the PHYSEQ object we already have by using the merge PHYLOSEQ to build it again from the scratch. The results should be identical and we can check it. So we write PHYSEQ1, merge PHYLOSEQ, and then PHYSEQ, sample data, random tree. Let's run our code. As you can see, we got the PHYLOSEQ class experiment, OTU table, 10 taxa, 10 samples, sample data, 10 samples by 2 sample variables, taxonomy table, 10 taxa by 7 taxonomic ranks, and phylogenetic tree, 10 tips and 9 internal nodes. And if we do it by making OTU table and text table that we have created, and then we put the same sample data and random tree, let's run it. So we got the same results. We can also check it by identical, so identical and then PHYLOSEQ1, PHYLOSEQ2. Let's run our code. We got true. Now let's build a couple tree plots with the new combined data. So we write plot tree, PHYLOSEQ1, color equal location, label tips, taxa names, and we put letterize left and plot margin 0.3. Let's run our code and we got our tree. So as you can see, different locations, A, B, C, D, each with the different colors and the sample names, OTU9, OTU10, OTU3, and so on. Now we can color it based on the depth. So we write PHYLOSEQ1 color equal depth. Then shape location, all the same. Taxa names, letterize, right, and plot margin 0.3. Let's run our code. As you can see, the higher the depth, the lighter the blue color, and the lower the depth, the darker the blue color. And we have here the location A, B, C, D, each with different symbol. So A is circle, B is triangle, and so on. We can also make a heat map, plot underscore heat map, and we put PHYLOSEQ1, run the code. As you can see, the lighter the color, the higher the abundance, the lower the abundance, the darker the color. And we have here the samples, and here is the OTUs, 1, 2, 3, and so on. And we can also add labels using plot, heat map, PHYLOSEQ1, taxa label, phylum. Let's run our code. As you can see, here are the samples, and instead of OTUs, we have the phylum, and we have our letters here. And now, what if we want to import the microbial community datasets into R for our analysis? In this case, we can use the microbiome _me QIIME function from the PHYLOSEQ package. This function was designed to interface with the QIIME database, which previously contained numerous microbial datasets. However, it's important to note that the QIIME database is currently down indefinitely, so this function is now primarily for reference purposes. The PHYLOSEQ package provides the capability to import various types of biome files seamlessly. When using the import biome function, we can also import related files, such as phylogenetic trees and reference sequences like FASTA files. So firstly, we define the file path, and in this case, this will be within the PHYLOSEQ package. So we use the special features of the system file command to get the path. This should also work on your system if you have PHYLOSEQ installed, regardless of your operational system. So reach dense system file and then ex data reach dense OTU table.biome package PHYLOSEQ, and reach sparse we put x data reach sparse OTU.biome and also package PHYLOSEQ in all package or PHYLOSEQ. And then minimum or min dense biome, we put x data, min dense OTU table, min sparse biome, and then tree file name, and then ref, sequence or seq, file name, equal system file, x data, biome ref seq dot fasta, package PHYLOSEQ. Let's run our code. Now we have defined the file path. Let's use this as argument to import biome function. Note that the tree and reference sequence files are both suitable for any of the example biome files, which is why we only need one path for each. In practice, you will be specifying a path to a sequence or tree file that matches the rest of your data, include tree tip names and sequence headers. So, we use import biome, reach dense biome, tree file name, refseq file name, parse function equal parse taxonomy, and then green genes. Let's run our code. As you can see, we got OTU table, 5 taxa and 6 samples, sample data 6 samples by 4 sample variables, 5 taxa by 7 taxonomic ranks, 5 tips and 4 internal nodes, 5 reference sequences. The OTU table 5, here 5 taxa and 6 samples, is identified in the dataset, representing a group of closely related organisms. This data has 6 different samples where these OTU were observed. And for sample data 6 samples for variables, this means that the metadata associated with 6 samples consisting of 4 different variables, such as environmental conditions or sample characteristics. And then 5 taxa by 7 taxonomic ranks. The taxonomy table provides information on the classification of the 5 OTUs across 7 different taxonomic ranks like domain, phylum, class, order, family, and so on. This allows for a detailed understanding of the biological classification of each OTU. And then 5 tips and 4 nodes for phylogenetic tree. The phylogenetic tree represents the evolutionary relationships between the 5 OTUs. It has 5 tips, the OTUs themselves, and 4 internal nodes. And lastly, DNA string set, 5 reference sequences. This indicates that there are 5 reference DNA sequences associated with the OTUs. We have explored the import biome function using the reach dense biome file in the previous code. Now we turn our attention to the reach sparse biome file. The main difference lies in how the data is stored. The reach sparse biome format contains many zeros, representing a sparse dataset where only non-zero values are recorded. This allows for more efficient storage, particularly in larger datasets with numerous absent operational taxonomic unit or OTUs. However, the handling of zeros in the sparse format requires careful consideration during the analysis. By using the sparse format, we can still import the necessary OTU table, sample data, taxonomy table, phylogenetic tree, and reference sequences. But we must be aware of the implications that missing data may have on our interpretations and results. So, we got import biome, reach sparse biome, tree file, refseq file name, parse function equal parse taxonomy greens or green genes. We can run our code, and here we have it. Now we will examine the use of the minimum dense biome file. The key difference with this file type is that it is a minimal dense biome file, which typically contains fewer samples or taxa compared to reach formats. This can result in a simpler dataset that may be easier to handle and analyze. So, we write import biome, minimum dense biome, tree file name, refseq file name, parse function equal parse taxonomy green genes. Let's run our code. And here we have it. Now we will look at the import biome function applied to a minimal sparse biome file. The minimal sparse format typically contains a limited number of taxa and samples, and it is designed to store data efficiently by only recording non-zero entries. So, we write import biome, minimal sparse biome, tree file, refseq file name, parse function equal parse taxonomy green genes. Let's run our code. And here we have it. In practice, we will store the result of our import as some variable name like my data. And then we will use this data object in downstream data manipulation and analysis. So we create a variable called my data and then equal to import biome, reach dense biome, tree file name, refseq file name, parse function equal parse taxonomy green genes. Let's run our code. And here we have it. Now let's visualize the phylogenetic tree using the plot tree function. This allows for comprehensive representation of the microbial community structure. This function enables us to customize the appearance of the tree by mapping different variables to colors, shapes, and sizes. So we write plot tree, and then we put my data that we have created, and then color genus, and then shape equal body site. Let's run our code, and here we have it. In the phylogenetic tree is a number located in the middles between the two branches, like here, or here, or here. It represents the branch lengths. These branch lengths are indicative of the evolutionary distance between the connected taxa. In simpler terms, they show how different or similar two organisms are in terms of their genetic makeup. A longer branch suggests a greater evolutionary divergence, indicating that the two taxa share a more distant common ancestor, while a shorter branch indicates that the taxa are closely related. So as you can see, we have genus and we have different types. Body site, gut or skin. The gut is circular shape, skin is triangular, and for abundance 1, 2, or 5. Now let's plot the richness in our data. However, before we do that, it's important to ensure that our data is free of any taxa with zero abundance. This step is crucial as it allows for more accurate representation of microbial richness in our plots. After filtering the data, we can proceed to visualize the richness of our samples based on different body size and their description. So, as we said, firstly we remove samples with zero abundance. My data underscore clean equal to prone samples when we put sample sums, and then my data. Firstly, we remove samples with zero abundance. My data underscore clean equal to prone samples, and then we put sample sums, my data, and we make it more than zero, and then we put my data, and then remove OTUs with zero abundance. We also do the same, but instead of prone samples, we put prone taxa. And then we plot the richness. Plot underscore richness, my data underscore clean that we have created, and we put the measures of the alpha diversity and color equal description, plus GMPox blot and theme classic, and then theme strip.background equal to element underscore blank. Let's run our code. And here we have it. Let's look at our plot. We have three plots, y-axis representing the alpha diversity measure and x-axis representing the body site. In each plot, we have box plots for gut and skin. So for example here, regarding the alpha diversity measure observed, it measures the total number of unique species present in a sample, reflecting species richness. So for example here, the gut, the observed OTU ranged from 2.5 to 3.5, indicating a rich diversity of unique microbial species in the gut. And for skin group, a single line at 2, this suggests low observed richness in skin microbiomes, with fewer unique species present. In the second plot, for SHA-1 index, it measures an estimate of species richness, particularly accounting for rare species that may not have been captured in sampling. For the gut group, the index ranges from 4 to 7.5, indicating high estimated species richness in the gut. But for the skin, the index from 2 to 2.5 indicates limited potential species richness in the skin microbiome. And lastly, Shannon index. It takes into account not only the number of unique species, but also how evenly they are distributed. In this case, for the gut, it's about 0.85 to 1.25, and for skin, it's about 0.7 to 0.75. Now let's visualize the composition of our microbial community using bar plot. By plotting the relative abundances of different genera, we can gain insights into the diversity and the structure of the microbial populations present in our samples. So, we write bar plot and then we put my data, fill, and genus. Let's run our code. As you can see, abundance and samples from sample 1 to sample 6 And here we have different types of genus. So as you can see, in sample 1 we have 2 types, in sample 2 we have 3 types, and so on. Now let's take a closer look at the reference sequences associated with our PHYLOSEQ object. The refseq function allows us to extract and examine the reference DNA sequences that correspond to the operational taxonomic units or OTUs in our dataset. So we use refseq and then we put our data. Let's run our code. As you can see, the output provides a DNA string set containing 5 DNAs. As you can see, 1, 2, 3, 4, and 5. So we have 5 DNA sequences. Each entry represents a different operational taxonomic unit or OTU in the dataset, along with many details. So for example, here for the first sequence, here we have a width of 334 base pairs, as you can see here. And for the second one, this one, we have about 465 base pairs, and so on. Now, what if we want to analyze microbiome data generated by QIIME, which is quantitative insights into microbial ecology? We can utilize the import QIIME function from the PHYLOSEQ package in R. This function allows us to import legacy output files, including an OTU file that contains both OTU abundance and taxonomic identity information. Additionally, a map file with sample covariates can be converted into sample data class used by PHYLOSEQ. QIIME might also generate a phylogenetic tree, which can be imported with this function or separately using read tree. So we put OTU file and then system.file xdata gpotu table rand short.text.gz and then package PHYLOSEQ and then map file system.file and we put xdata master map.text and then tree file xdata gp3 rand short newick.gz and then rs file xdata reference sequence.fasta and then QIIME data import QIIME OTU file map file tree file rs file. Let's run our code and here we have it. So in this code snippet we are importing microbiome data generated by QIIME into R using import QIIME function from the PHYLOSEQ package. For the OTU file, we load gzipped OTU table file which contain OTU abundance and taxonomic identity. And then for map file, we load a sample metadata file which includes covariate information for the sample. And for the third one, tree file, we load the gzipped newick formatted phylogenetic tree file that represents relationships between OTUs. And lastly, rs file. Here we wrote the reference sequence file in FASTA format, which contains the reference sequences for the OTUs. And now, let's print the QIIME data. As you can see, we got the OTU table, sample data, text table, phi tree, and reference seq. We can also plot it on bar plot. We just write plot bar and the QIIME data, x equals sample type, and fill with phylum. Let's run our code. As you can see, so we have actinobacteria, OP9, planktomycetes, cyanobacteria, chloroflexi, and others. And as you can see here in the x-axis is the sample from tongue or soil, skin, ocean, mark, or abundance on the y-axis. We can also generate a heat map by using plot heat map function. So plot heat map and then QIIME data, sample.label, sample type, species.label, phylum. Let's run our code. And here we have it. As you can see, here is the samples and here is the OTU. And regarding the abundance, the darker the color, the lower the abundance, and the lighter the color, the higher the abundance.